All right, this one's going to be a quick overview of Chapter 7, which is on membrane structure and function. So, the key things that you want to take away here is first and foremost, what does the cell membrane do? And it's going to basically be kind of a barrier in terms of allowing specific things in and out. I'm going to primarily be talking about eukaryotic cells in this video because a lot of this has to deal with specific proteins that are going to help let things in and out. But regardless, whether it's prokaryotic or eukaryotic, the structure is really just made up of a phospholipid bilayer. So in terms of that, let's kind of get into what is a phospholipid, right? So this whole thing is going to be essentially the polar head. And again, if we say it's polar, it typically is going to have some form of a charge. And all of this right here is going to be your nonpolar tail. So one of the key components is how do you identify if it's nonpolar or not? If it's primarily made up of a hydrocarbon, it's referring to it as being nonpolar. So right here, this is a relatively lengthy nonpolar tail. I have one double bond here, so this makes it what's considered unsaturated, where this one up top is, I would say this one is going to be saturated. This one down here is unsaturated. I'll talk about that in a minute if you forgot. But this is your phosphate head, right? There's your phosphate group. That's what gives it this really polar charge. And again, the two nonpolar tails like to orient themselves together, and this is what's going to help make up your cell membrane. So you get your polar heads here, your polar heads over here, and everything in the center is nonpolar. So all of that nonpolar or hydrophobic components is going to be making up the interior of that membrane. Other than that, a couple of reminders of this is going to be referring to phospholipids are what are going to be considered amphipathic. That means it has both a polar region and a nonpolar region. Yeah, that's pretty straightforward. So in terms of this, you do want to make up and understand kind of what each one of these macromolecules is involved with. The lipids is going to give it that hydrophobic region. There's proteins that are going to be embedded within the cell membrane, as well as carbohydrates that allow cell-to-cell recognition. So looking here, each one of these things in purple are going to be the proteins, so I'll just abbreviate those as your proteins. Here's going to be your carbohydrate component that can actually allow cell-to-cell recognition. You can also get some cholesterol embedded in the cell membrane that allows for some temperature fluctuation to not happen. So this would essentially be inside the cell. This would be outside your extracellular matrix there. So now, in terms of this, I always kind of like to remind you guys of asking you, well, from the previous video, if I asked you if these are proteins, well, where did these proteins come from? Well, remember, you had your nucleus in here. The nucleus helped make your mRNA. You made the protein. It used that endomembrane system to actually get to the cell membrane, okay? So other key things on this, in terms of the fluidity, that's what I was just mentioning earlier. In terms of cholesterol, it can act as a temperature buffer. It makes it a little more challenging to actually change temperature. In terms of that, for the most part, cells have to have some type of flexibility. And this is just kind of showing you a silly little meme where if it's polar, like the polar head, it wants to be next to water, which water for the most part is relatively extremely polar actually. So it's always going to be basically that orientation that if you go back and take a look here, these polar heads are facing towards the polar interior because remember cells are primarily made up of water. Same thing on the exterior, the polar heads are facing the polar solution that they're in. And there's actually a really weird FRQ I think I'm going to give you guys. If not, I'll sign it again. That the AP test can kind of flip that script a little bit. So this is just showing you types of fluidity. If there's these gaps, these things are on, these little tails are going to be unsaturated. That gives it that little tiny kink that allows for increased fluidity. This is showing cholesterol. It's nonpolar. It can be embedded within the nonpolar region, right? This whole region right here is nonpolar. If it's super saturated in the sense that your nonpolar tails are saturated with hydrogen, it makes it really, really firm. Other than that, key things about some terms that you should know is integral versus peripheral. Integral is going to be a protein that's going to span, for the most part, the interior of the cell membrane, Or if it's peripheral, I would abbreviate this one right here. Peripheral are just going to be one of those surface proteins, so you just see it on the surface. Now, most of the proteins that are going to be in the cell membrane are going to be integral, so they're going to span all or part of that membrane, referring to the interior of it. So this is one thing that I want to take a moment and actually make sure you guys feel okay with. So this whole thing in purple, this thing right here is your protein, and it's folded. It looks great, but I want to remind you guys that all of these amino acids right here internally, all of these, remember, are going to be polar, because polar amino acids want to be next to polar amino acids, and they're essentially hiding themselves. Now, if I looked at these amino acids that are essentially allowing it to anchor within the cell membrane, these amino acids right here are going to be non-polar. That's allowing it to actually anchor itself within the cell membrane. So you just want to understand that different parts of the protein are going to have polar properties, different amino acids are going to be non-polar, but it's those non-polar ones that are going to allow it to actually anchor within the cell membrane itself. So if we were realistically giving you an example of this, if this is, let's say there's a channel here or an opening that's polar, if I had a polar molecule, it would actually be able to diffuse through that polar region and get into the cell itself. So just understand that that's how these proteins are going to be embedded within the cell membrane. Now, other than that, there's a lot of other functions of proteins that are going to be within the cell membrane. Key things that we see a lot is going to be transport, specifically in signal transduction pathways as well. But it can also do things like enzyme activity, cell recognition, as well as the cellular joining, like I talked about in chapter 6. But like I said, the bulk of it is going to be for transport. Cell-to-cell recognition is important, just to remind you guys that this is showing you a carbohydrate right here that's allowing it to recognize an adjacent cell nearby. This is showing you enzymes, but I like to put a little asterisk here because a lot of this is going to be specific to chapter 11. The signal binds to the receptor protein, and that starts the cell signaling process. Now, other than that, if you don't know these terms, glycolipids, glycoproteins, remember that glyco is a prefix to just a carbohydrate, right? So glycogen is one of those polysaccharides. So it's literally just glycogen attached to either a lipid. We call it a glycolipid. If it's glycogen attached to a protein, we call it a glycoprotein. But regardless, it's going to allow it to recognize adjacent cells. So in terms of key things about membrane transport, absolutely, massively important to differentiate between these two. Passive transport is talking about no energy required. It's always going down its concentration gradient. And when we say down its concentration gradient, you're going from high to low. Other key components about this is that there's no ATP involved. And active transport obviously is going to be requiring ATP or GTP depending on the type of scenario they give you. You're going against the concentration gradient, so you're literally pushing things from a low concentration to a higher concentration. It's not naturally going to want to do that, so that's why it does require some ATP. Now, other key things about passive transport that some students get confused with. A lot of people understand that if it's passive, it doesn't require ATP, but it still can require a protein. It just depends on the type of passive transport. So this is just showing you kind of what going down its concentration gradient would be in. So right, if I let this loose, those molecules are going from a concentrated area to a not so concentrated gradient. So I like this little simulation. I think you guys have all seen this in class, right? So it's pretty equal, but if we follow this little red molecule, that's a non-polar molecule. It can sneak right through the cell membrane, right? So if you look through all of these, again, you just follow through. It's non-polar. It can just sneak right through the cell membrane. It doesn't require protein. But this blue molecule, relatively large and polar, it's not going to want to go through. So kind of simple visual to kind of help you guys out there. But in terms of passive, right? So there's a couple of key ones you have to know in terms of passive transport. It's allowing things to go down their concentration gradient from high to low. No energy is required. And specifically, one of the key things here is that there's going to be no, if we're talking about simple diffusion, So you're moving down the concentration gradient, no energy required. And it really doesn't even require a protein. So things can just literally diffuse right through. So things that are nonpolar and relatively small can just literally slip right through the cell membrane. So understand that. And if we talk about facilitated diffusion, we're still most definitely going to call this passive transport, right? Because it doesn't require ATP. No energy is required. But it does require some of those channel proteins, right? And so these proteins, the key thing is that each protein is specific to what it's letting pass through. So each protein is specific to what it passes through, to what goes through. And so basically what we're saying here is like, let's say that if this is a specific polar molecule, it's going to require this specific channel protein that's allowing it to go down its concentration gradient, right? So these things are all diffusing down their concentration gradient, right? This would be a non-polar molecule. It's showing that these molecules can actually still diffuse down, but no protein is needed. So that would be an example of your simple diffusion, right? So this one on the left is simple diffusion. This one is showing facilitated diffusion. So think of it as like the protein is facilitating that diffusion of that molecule. It's helping it. So again, key thing is that each protein is specific to what it lets go through, right? Aquaporins would be an example too. Water is polar, which is kind of a trip that you would think that water can just freely pass back and forth. It can in a weird way without a protein, but for the most part, if you're getting large amounts of water, into a cell, it requires what's called an aquaporin. Carrier proteins do essentially the same thing as a channel protein, except for they have to actually change shape throughout the duration of it. So this is just showing you an example of a carrier protein right here, that where the molecule binds to it, it changes shape, and it lets that solute pass through. So a nice little visual comparing just facilitated diffusion between a channel protein versus a carrier protein. But key thing is that they're still letting molecules go down their concentration gradient from high to low. No ATP is required. Now, one other key component about this chapter is about osmosis and where does water go, right? So in terms of this, you for sure have to know these terms: isotonic, hypertonic, hypotonic. Isotonic, where there's no net movement of water. So if we think about in terms of plant cells, if they're flaccid, water is equally in concentration on the outside. Animals' cells, for the most part, want to be in an isotonic state. So equal solutes on the outside as on the inside. And one thing that helps, at least even for me still, is that water goes to where there's more stuff, aka solutes. So water always goes to where there is more stuff. And I'll put quotations around that because the appropriate term for stuff is just solutes. So water moves to where there's more solutes. And again, solute can be anything from calcium ions to proteins. So wherever there's more stuff, that's where water typically wants to go. So if we're thinking about an isotonic cell, there's equal stuff on the inside and the outside. Water's not moving in one particular direction. If we think about a hypertonic state, water is moving out. And essentially, when you think about, if it's hypertonic, the solute concentration is greater on the outside, right? So if we had a cell and this is on the exterior and I have all these solutes, water is essentially gonna want to go out to where there is more stuff, okay? And the way I remember this compared to hypertonic, hypotonic, right? If there's hypotonic, if there's a bunch of solutes here and there's water on the exterior, and only a few solutes, but there's a lot of solutes on the inside, water is going to want to rush to where there is those more solutes, okay? So remember hypertonic versus hypotonic. The silly way that I remember this is hypo kind of sounds like hippo. Hippos are really, really fat. So if you think of hypotonic state, the cell gets really, really fat. Hypertonic, if you think of somebody moving all the time, they're typically really, really skinny. So really, really silly, but whatever helps you guys remember that. So in terms of that, this is just showing you a couple of representations, right? So this is showing you that water has left these cells, and they've essentially plasmolysed. So this is just showing you that there's some type of solute going on. So really, really important picture here, that if it is hypotonic in an animal cell, it's not good, because as water rushes in, the cells can actually burst. However, plant cells actually like to be hypotonic because they have the cell wall. It allows that pressure to get built up. So they actually like to be in a hypotonic state. So the more water they have. And that's partially in terms of this large center vacuole that those cells have. Isotonic, equal movements in and out. This is essentially where plant cells, but animal cells are essentially their happiest. Plant cells, they aren't loving that. And then neither plant cells or animal cells like to be in a hypertonic state where there's more solutes on the outside. So moving forward, this is just showing you a nice representation that these little purplish spheres are representing water, and these larger pinkish spheres are talking about solute. So relatively equal on the in and out. So no net movement. So water is going to be equally going back and forth. That would be an isotonic state. Now, if you look here, this is going to be a hypertonic solution. So if you look here at this visual, all of these are going to be your solutes. So let's say you have more stuff on the outside of the cell. It's showing you that water is going to want to move out. And this cell will definitely shrink, because it essentially is, there's more solutes on the outside, you're causing water wanting to leave. And we can relate this to water potential as well, which I will do here shortly. Hypotonic, there's more solutes on the inside. Remember that this is showing you all of your solutes, so all of your water is essentially going to want to rush in. So in terms of that, I think that is pretty straightforward, but this just kind of reminds you guys that cell walls help maintain that water balance, and it gives it that turgor pressure. Other than that, last really simple comparison. This one is just showing you simple diffusions, right? These purple molecules can just diffuse down. This is showing you a nice little channel protein that's allowing these molecules to go down their concentration gradient. And this is showing you a carrier protein. But all of these on the left side are going to be essentially passive transport. So they can simply just diffuse down. No ATPs required. They're going from high concentrations to low concentrations. Now, as we get into active transport, key things is it requires energy. So you're pushing things against the gradient that they normally do not want to. Now, one thing that you would see this a lot with is the electrochemical gradient, specifically when you think of cell respiration, right? So if I have a cell membrane here and I have a bunch of hydrogen ions on one side, when we think of energy in active transport, a lot of us are going to assume that it's ATP or even GTP. But any form of energy, we're going to require that as being active transport, right? So if I had, let me change this color up real quick. So if I try to push this proton, if this is my cell membrane here, and I'm trying to push this proton going from a lower concentration to a higher concentration, it takes some energy. So this is just reminding you, right? If you think of like this cell respiration, the electron transport chain, key thing that I just need you to understand is that when we think of electrochemical gradient, you're building up a gradient of these protons, and it's just showing you that it takes energy to push that hydrogen ion against the gradient. We also see this a lot when we think of the sodium-potassium pump, which we now do not have to cover, but really key thing, if it requires energy, it's going to be considered active transport. So just wanted to kind of put this one out there, because I think if they do test you on active transport, this might be one that they'll sneak in. You're pushing protons against the gradient, and the energy in this case is coming from the electron. So one of the other ones that get a little bit confusing is co-transport. So let me show you this visual. Right, step one, you take ATP, you're pumping those protons against the gradient, and then you build up this electrochemical gradient over here on this side. So let me try and just highlight this real quick. So if you're looking here, this is step one. You're using ATP, you're pumping those protons from a low concentration all the way over to a high concentration. So you're using this protein to pump that proton, and again, protons are those little hydrogen ions, against their gradient. You build up that gradient, so now all of a sudden you have a high concentration of these protons. These protons are now going to just do passive transport, where they're now going to be using one of these proteins. Those protons go through this protein. As they diffuse down, they almost literally force sucrose, which is this example here where there's a low concentration of sucrose, high concentration of sucrose on the inside. It's literally just forcing all of this sucrose against the concentration gradient. So it's kind of intense, but if you look at it, the ATP is using the protons, pushing the protons against their gradient. These protons get built up, and then as they diffuse back down, it pushes sucrose against the gradient. So really, really interesting way of using some form of active transport in this case. So crazy ideas here, but hopefully that makes some sense to you in terms of co-transport. It's really, the point is really you're just building up all these protons just to get those protons to diffuse back down. As they diffuse back down, they're pushing something else that's going against their concentration gradient. In this case, you're pushing sucrose from a low concentration to a high concentration, but you're not using ATP directly. All right, last bit is exocytosis. This is kind of a build off of last chapter, right? I talked about in the last video that it takes some ATP to push these proteins outside of the cell. That's going to be referred to as exocytosis. When you get to endocytosis, endo means going into the cell. So into the cell, we got three types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. So in terms of this, if we're doing phagocytosis, the solid particle just essentially buds into the cell and it steals a part of the cell membrane. If you think of pinocytosis, that's essentially where it's going to be uptaking liquids. So that's one way to remember the pinocytosis as it's taken in liquid. Pina colada, well, I shouldn't say this, but in terms of that, it's a liquid. So easy, simple. I think it was in one of my review books in college that you're taking in a liquid. That's how you're remembering that. Receptor-mediated endocytosis, this is just showing you that if this is a receptor protein right here, it is specific, right? So this by far is the most specific type. Depending on the type of receptor protein, it binds to the specific molecule, and then that molecule is now inside the cell. So just know that you're letting things inside the cell via endocytosis. Last bits of chapter 7. I'll make a separate video about water potential, but this is just kind of a revisitation from the POGIL that we did weeks back. But it's kind of a good representation of just kind of showing you if this is the protein, right? This would essentially, all these amino acids on the exterior that are actually anchoring itself, these ones would be non-polar amino acids. And the ones on the interior are going to be, those R groups are going to be polar. Yeah, just wanted to kind of point that one out. So that's kind of the end of